Hi, my name is Hao Sheng, and I'm going to present a interesting paper from the Cell, titled with molecular and spatial signatures of mouse brain aging at single cell resolution. As we aging, the brain naturally undergo the changes and leads to cognitive decline, motor issues, and increase the risk of disease like Alzheimer's. But the brain is made up of many different kinds of cells like neurons, glial cells, immune cells, and vascular cells, and we don't totally understand how of each of each of these cells respond to aging. Most previous studies have used bulk RNA-seq, which is averages gene expression across many cells. That means we are losing two things: the cell type's specificity and spatial context. We do not know exactly which cells are changing and where are they in the brain. So this study tried to fix that by combining two powerful technologies, which is single nucleus RNA sequencing and MERFISH, which is a spatial transcriptomics method. This lets the researchers studying aging at the level of individual cells, which are which can also let them see the cells area in the tissue. So what are the authors trying to figure out? Their hypothesis is that aging doesn't affect the brain cells in the same way. In particular, they think the glial cells and immune cells might undergo unique changes based on their location in the brain. So to test this hypothesis, they use two techniques. The figure 1A shows the overall design, and they study the three age groups of mice: the juvenile, adults, and aged mice. They focus on the frontal cortex and striatum. They perform single nucleus RNA-seq on about 80,000 cells to determine cell types and genes expression profiles. Then in figure 1B, they use MERFISH to spatially map the 416 genes across like 400,000 cells. In this way, they can link the transcriptional changes and to physical locations in the brain. And here's a simplified view of what they did. They first dissected the brains of three different age groups of mice and collect the tissue from the cortex, and then they run the single nucleus RNA-seq to classify the different cell types and determine which genes are expressed. And after that, they selected 416 genes based on the sequencing results and used MERFISH to map their spatial distribution in tissue slices. Finally, integrate both data sets to build a high resolution spatial map of aging mouse brain. Figure 2A shows that neurons stay relatively constant across age, but non-neural cells, especially glia cells, change a lot. In figure 2B, we can see a new subtype of oligodendrocytes called oligo3. Uh, they appear only in aged mice. These cells express genes like C4b, which are associated with inflammation. So aging doesn't kill neurons. Instead, they transform glial cells to new cell types that are more immune-like and inflammatory. Now, looking at figure 3C to 3F, these spatial maps show where different glial subtypes are located. You can clearly see the oligo3 and astro2, which are inflammatory subtypes, are concentrated in the white matter. So glial cells activation is not spread evenly. They localized in a specific location in the brain. This means the white matter might be the region specifically related to the brain aging. And figure 4A shows that most age-related gene expression changes occur in non-neural cells. In figure 4B and 4C, we see that these genes are enriched of immune-related functions like antigen presentation and some signaling. So even without infection, aging can trigger the glial cells to adopt an immune-like state. To quantify how activation of these glial cells, they compute the activation score. The figure 5A and 5B shows that activation is highest in the corpus callosum. And figure 5C, we can see the spatial clustering of activated glia. So the activation often happens near the vascular associated cell. This indicates the glial activation might be driven by location microenvironments, not just internal aging signals. To test whether aging is just chronic inflammation, so the researchers compared aged mice with young mice and treated with LPS, which is a compound that induced the inflammation. In figure 6C to 6E, they show that there is some overlap in gene expression in aging and the inflammation. But the aging has distinct signature that is not identical to LPS. So aging mimics the inflammation in some ways, but it's not the same. Finally, aging mainly affect the glial cells, uh, and these changes happen in white matter regionally, and spatial single cell analysis can help to discover these patterns. But these studies have some limits, like they only look at the two brain regions and not functional validation yet. They didn't test the cost effect relationships. And this is the reference. So thank you for listening, and I hope this can give you a better understanding of how brain ages and how these tools can like Murfish and single nucleus RNA sequencing can helping to study in more detail.